There is a surface, white and still. Wind moves across it in patterns that leave no trace. The sky above holds no warmth. The ground beneath offers no softness. Everything is pale. Everything is cold. The ice extends in all directions without variation. No plants break through. No animals leave tracks that last. The silence is not peaceful. It is total. Beneath your feet, the world continues downward. Not into soil. Not into rock. Into more ice. Kilometers of it. Pressing down with a weight that has no comparison in ordinary life. The ice is not transparent. It is not blue. It holds bubbles of air from atmospheres that no longer exist. It holds dust from continents that have shifted. Time is compressed into layers you cannot see. And below all of it is something else. The ice sheet is not thin. In places, it reaches depths of 4 kilometers. That is the height of mountain ranges in reverse, stacked vertically into darkness. The pressure at the bottom is immense. The temperature should be far below freezing. But something changes the rules down there. The ice does not continue forever downward. At a certain depth it stops. Not because it melts in the way you understand melting, but because conditions become different. There is a boundary. On one side is ice that has not moved in millennia. On the other side is water. Liquid water. In darkness. Under pressure that would crush most vessel designed by humans. The water does not freeze. It does not boil. It simply exists in a state that should not be possible. You cannot reach these places by drilling through surface ice. The distance is too great. The equipment required does not exist in practical form. The risk of contamination is absolute. What lies beneath has been separated from the surface world for lengths of time that exceed human civilization. Some of these waters have been isolated for millions of years. They have never known sunlight. They have never exchanged gases with the atmosphere above. They have developed in complete separation from the biological systems you understand. Your intuition about water does not apply here. Your assumptions about life do not transfer. The rules are different. Not slightly different. Fundamentally different. And yet the water remains. Still. Dark. Waiting. There are over 400 of these places beneath the ice. They are not ponds. Some are larger than seas you can visit. They connect to each other through channels you cannot see. They form systems that operate without light or wind or seasons. What exists in them remains mostly unknown. What processes shape them cannot be observed directly. But evidence emerges slowly. Through ice cores. Through radar surveys. Through instruments that measure gravity and magnetism. The picture builds slowly. A map of hidden water. A network of lakes in perpetual night. This is where the descent begins. Welcome to Info Jungle. If you enjoy calm, thoughtful documentaries about the natural world, consider subscribing. These videos are created to help you relax while learning at a gentle pace. The first clue came from radio waves. In the late 1960s, researchers began using radio echo sounding over Antarctica. The technique sends radio pulses down through ice. When the pulses hit different materials, they bounce back. Ice returns one kind of signal. Rock returns another. Water returns something distinct. The reflections are strong and bright. In certain regions, the returning signals showed something unexpected. Beneath kilometers of solid ice, the radar detected flat surfaces. Perfectly flat. Smoother than the ice above. Smoother than the bedrock around them. The only material that creates surfaces this flat is liquid water. At first, the findings were treated with skepticism. Water beneath that much ice seemed impossible. The temperature at those depths should be far too cold. The pressure should force any water into exotic forms of ice. But the signals were clear, and they appeared in multiple locations across the continent. Over time, the evidence became undeniable. There were lakes beneath Antarctica, hundreds of them, sealed away under ice that had not melted in millions of years. The first confirmed lake was named Vostok. It lies beneath Russia's Vostok Station in East Antarctica. The lake's surface sits under more than 3.5 kilometers of ice. The lake itself is enormous, roughly 250 kilometers long, 50 kilometers wide, in some places over 1,000 meters deep. It is one of the largest lakes on Earth, and it has been hidden in total darkness for at least 15 million years. Ice is not empty space. It is not light. It is water in solid form compressed over thousands of years. The Antarctic ice sheet holds enough frozen water to raise global sea levels by nearly 60 meters. That mass does not rest gently on the continent. It presses down with enormous force. At the surface, the pressure is normal. 1 atmosphere. The same pressure you experience standing at sea level. But pressure increases with depth. Every 10 meters of ice adds roughly 1 additional atmosphere of pressure. By the time you reach 3 kilometers down, the pressure has increased to 300 atmospheres. That is 300 times the pressure at Earth's surface. At those pressures, strange things happen to ice. The crystal structure changes. Ice becomes more dense. It can flow like a very slow liquid. The base of the ice sheet is not rigid. It deforms over time under its own weight, moving outward from the center of the continent toward the coasts. This movement creates friction, and friction creates heat. Not much heat, but enough to matter. At the base of the ice sheet, temperatures rise just enough to reach the melting point. Water forms, not because the environment is warm, but because the pressure changes the temperature at which ice melts. Under 300 atmospheres of pressure, ice melts at temperatures several degrees below 0 Celsius. The water that forms is supercooled by surface standards, but liquid nonetheless, trapped between ice above and rock below, unable to freeze, unable to escape. Not all subglacial lakes are the same. Some are small, a few kilometers across, shallow pools caught in depressions in the bedrock. Others are vast. Lake Vostok is the largest, but not the only giant. Lake Whillans sits beneath the West Antarctic ice sheet. It is smaller than Vostok, but still substantial. Roughly 60 square kilometers in surface area, 20 meters deep in places. It has been studied more closely than other subglacial lakes. In 2013, researchers drilled through the ice above Lake Whillans. They used sterile techniques to avoid contamination. The drill took days to penetrate the ice. When it finally broke through into the water below, samples were collected immediately. The water was cold, just below freezing, but it was liquid, and it was not sterile. The samples contained life, bacteria and archaea, microorganisms adapted to conditions that seem hostile to biology. No light, no oxygen from the surface, extreme pressure, limited nutrients. Yet they had survived. And not just survived, they had formed functioning ecosystems. The discovery confirmed what some scientists had suspected. Life does not require sunlight. It does not require warmth. It requires only liquid water and energy. And beneath the Antarctic ice both exist. The lakes are not isolated. They connect to each other. Beneath the ice sheet, water flows through channels carved into the bedrock. Some channels are small. Others move significant volumes of water. The flow is not constant. It changes over time as pressure shifts and ice moves. Some lakes drain suddenly, releasing water downstream to other lakes. The surface ice above these lakes can drop by meters when this happens. Satellites detect the subsidence from space. The movement is slow by human standards. It takes months or years. But it is real. The subglacial system is dynamic. Water accumulates in some basins. It drains from others. The balance shifts continuously. Lake Whillans is part of a network that extends hundreds of kilometers. Water flows from higher elevations near the center of the ice sheet. It moves gradually toward the coast. Along the way, it fills depressions in the bedrock. These become lakes, temporary or permanent, depending on the local conditions. Some lakes have existed for millions of years. Others may be only thousands of years old. The system as a whole is ancient, but its details change. The map of subglacial Antarctica is not static. It is a living hydrology, shaped by forces that operate on timescales longer than human history. Water in subglacial lakes is not pure. It carries dissolved minerals from the bedrock below. It contains particles of rock ground down by the moving ice. It holds gases trapped from ancient atmospheres. The chemistry varies from lake to lake. Some lakes are relatively fresh, low in dissolved salts. Others are more concentrated. The water in Lake Vostok is extremely fresh, similar to distilled water. This suggests limited interaction with the bedrock. The lake may be sealed off by sediment layers, or the bedrock itself may be chemically inert. Other lakes show higher mineral content: calcium and magnesium from carbonate rocks, silica from granites and basalts, iron from volcanic deposits. Each lake reflects the geology beneath it. The bedrock of Antarctica is complex. Ancient mountain ranges buried under ice, rift valleys filled with sediment, volcanic regions still showing thermal activity. All of this influences the water above it. The chemistry of a subglacial lake is a record of its interaction with the solid earth. In some lakes, geothermal heat rises from below. This heat comes from the planet's interior. Radioactive decay in the mantle generates warmth. In certain regions, this heat flux is higher. Volcanic activity adds additional energy. The heat does not warm the water significantly, but it prevents freezing at the base. It drives slow convection currents in the water. The discovery of life in Lake Whillans changed assumptions. For decades, scientists debated whether subglacial lakes could support biology. The environment seemed too extreme. No light means no photosynthesis. No photosynthesis means no base for a food chain. But life found another way. The microorganisms in Lake Whillans do not rely on sunlight. They use chemosynthesis instead. Chemical reactions provide energy. Minerals dissolved in the water serve as fuel. Iron and sulfur compounds are oxidized. The reactions release energy that cells can use. This is the same strategy used by organisms in deep-sea hydrothermal vents and in caves kilometers underground. Wherever liquid water and chemical gradients exist, life finds a way to persist. The microbes in subglacial lakes are not fast-growing. Their metabolisms are slow. Division times measured in days or weeks rather than hours. But they are resilient. They have adapted to near freezing temperatures. They can function under extreme pressure. They survive in darkness. They extract nutrients from environments that seem barren. The diversity is limited compared to surface ecosystems. But it is real. Different species occupy different niches within the lake. Some metabolize iron. Others process sulfur. Some break down organic carbon that filters down from above. Together they form a functioning ecosystem. Simple, slow, invisible from the surface, but alive. 15 million years is not a number that translates easily. It exceeds the span of human civilization by thousands of times over. It predates the existence of recognizable human ancestors. During that time, the surface world changed completely. Ice ages came and went. Continents shifted position. Ocean levels rose and fell by hundreds of meters. Species emerged and vanished. But beneath the ice, the lakes remained. Separated, unchanging in their separation. Lake Vostok has been sealed for at least this long. Perhaps longer. The ice above it accumulated slowly over millions of years. Snow fell on the surface, year after year, century after century. Each layer compressed the layers beneath. The weight increased gradually. Eventually, the ice became thick enough to isolate the lake completely. No exchange with the atmosphere. No input of organic material from above. No genetic exchange with surface ecosystems. For 15 million years, the organisms in Lake Vostok have evolved in complete isolation. Every generation separated from the outside world. Every adaptation shaped only by conditions within the lake. This is a timescale that makes comparison difficult. Your lifetime is roughly 80 years at most. Human civilization is perhaps 10,000 years old. Modern humans as a species are 300,000 years old. Lake Vostok has been isolated 50 times longer than that. The organisms living there today are descended from ancestors that entered the lake before Antarctica was fully frozen, before the ice sheet reached its current size, when the continent still had regions of open water and exposed rock. Those ancestral organisms became trapped, and everything that followed happened in darkness. On the surface, seasons exist. Summer brings continuous daylight. Temperatures rise slightly above freezing in coastal regions. Ice melts at the edges. Meltwater flows in streams. Algae blooms in temporary ponds. Winter brings darkness. Temperatures fall to -70 Celsius or lower. All liquid water freezes solid. Biological activity ceases. The cycle repeats annually. Life on the surface is shaped by this rhythm. Breeding times, migration patterns, dormancy periods. Everything is timed to the predictable return of warmth and light. But beneath the ice, there are no seasons. The temperature does not change with the passage of months. Light does not return in summer. The water temperature remains constant, just below freezing. Always. There is no cue for reproduction based on seasonal warmth. No migration triggered by changing day length. No dormancy required for winter survival. The environment is static, not in the sense of being dead, but in the sense of being uniform across time. One day is identical to the day before. 1 year is identical to the previous year. 1,000 years passes without detectable change. For organisms living in this environment, time loses meaning. There is no advantage to tracking seasons. No benefit to anticipating environmental shifts. The selective pressures are different. Survival depends on efficiency, on minimizing energy expenditure, on tolerating constant cold, on extracting resources from an environment that provides nothing new. The pressure at the bottom of the ice sheet does not fluctuate. It does not ease during summer. It does not increase during winter. 300 atmospheres. Constant. Unrelenting. At the surface, you experience 1 atmosphere of pressure. The weight of the air above you. You do not notice it because your body is adapted to it. Fluids inside your cells are at the same pressure as the air outside. If you ascend to high altitude, the pressure decreases. Your body compensates. Blood vessels dilate. Breathing rate increases. If you descend underwater, the pressure increases. At 10 meters depth, the pressure doubles. Your body compresses slightly. Air spaces in your lungs and ears feel the change. At 30 meters, the pressure is 4 atmospheres. Nitrogen begins dissolving into your blood. If you ascend too quickly, the nitrogen comes out of solution too fast. Bubbles form in your tissues. This is decompression sickness. It can be fatal. At 300 atmospheres, the rules change entirely. Gases behave differently. Proteins in living cells change shape. Chemical reactions that work at surface pressure fail. The organisms in subglacial lakes are not visiting this pressure. They live in it permanently. Their cells are adapted to conditions that would destroy surface organisms instantly. The proteins in their cell membranes are structured differently. More rigid in some areas, more flexible in others. They maintain function under compression that would denature normal proteins. Their metabolic pathways operate at rates that seem impossibly slow. But speed is not relevant here. There is no competition for rapid growth. No predator that can be outrun. Only the constant work of maintaining cellular integrity under pressure that never relents. You cannot see into these lakes. Not with cameras. Not with probes. Not without drilling through kilometers of ice. And drilling carries risk. Every hole through the ice is a potential contamination pathway. Surface microbes could be carried down. Drilling fluids could enter the lake water. The pristine chemistry could be altered. The isolated ecosystem could be disrupted. This is not a theoretical concern. It has happened. Early drilling attempts were less careful about sterilization. Some boreholes used antifreeze to prevent refreezing. Some used fuels and lubricants that are toxic to microbial life. These substances remain in the ice, slowly diffusing downward over decades. When Lake Vostok was first reached by Russian drilling teams, the decision was made to stop just above the lake surface, to not penetrate the water. The borehole was sealed. The lake remains unsampled directly. Everything known about Lake Vostok comes from indirect methods: radar imaging, seismic surveys, ice core analysis. The ice directly above the lake contains frozen lake water. This ice accreted from below. Water that touched the bottom of the ice sheet froze, slowly building up over thousands of years. When ice cores are drilled, these frozen lake water layers can be identified. They contain clues: trapped gases, chemical signatures, sometimes microbial DNA. But the clues are fragmentary. The picture they provide is incomplete. Most of what exists in the lake remains unknown: the size of the microbial population, the diversity of species, the structure of the food web, the rate of reproduction. All of this is estimated, guessed at, inferred from limited data. The lake exists. That much is certain. What lives in it is largely a blank space in scientific understanding. In normal lakes, water moves. Wind creates surface currents. Temperature differences drive convection. Warm water rises. Cold water sinks. Rivers flow in. Streams flow out. The water is replaced over time. Nutrients enter from the surrounding landscape. Oxygen is replenished from the atmosphere. The lake is connected to the larger hydrological cycle. But subglacial lakes are not connected. They are endpoints. Water enters slowly from melting ice. Water leaves slowly through outflow channels. But the exchange rate is extremely low. In Lake Vostok, the average water molecule may remain in the lake for thousands of years before exiting. There is no wind to drive surface mixing. The ice above is motionless relative to the water. Temperature differences are minimal. The entire water column is within a few degrees of freezing. Convection is weak. In the deepest parts of the lake, water may sit undisturbed for millennia, never mixing with the layers above, never exchanging dissolved gases, never receiving fresh nutrients. This is stagnation in the most literal sense. Not stagnation as decay, but stagnation as stillness. Water that has not moved in a time longer than recorded history. Oxygen levels in these deep zones are unknown. They may be extremely low or completely absent. If no oxygen reaches the deep water, then only anaerobic organisms can survive there. Bacteria that do not need oxygen. Archaea adapted to conditions where oxygen is toxic. These organisms would be even more removed from familiar life. Their metabolisms running on chemical reactions that produce no oxygen as waste. Their growth rates even slower than the aerobic microbes in shallower zones. Invisible, undetected, possibly undetectable with current methods. At the bottom of most lakes, sediment accumulates. Particles settle from the water above. Organic material, mineral grains, dead organisms. Over time, these build up into thick deposits. The sediment becomes a record of the lake's history. Layers can be counted like tree rings. Each layer represents a period of time. Changes in sediment composition reveal changes in the lake environment. But in subglacial lakes, the sediment is different. There is no organic rain from above. No leaves falling from trees. No pollen drifting down. No dead plankton sinking through the water column. The only particles are mineral grains, ground from bedrock by the moving ice sheet. carried into the lake by meltwater, or eroded from the lake floor by slow currents. The sediment accumulates slowly, millimeters per 1,000 years. In some lakes, the sediment layer may be only a few meters thick. In others, it could be tens of meters. But thickness is not the issue. The issue is what the sediment contains. In Lake Willen's sediment samples showed high microbial activity. The mud at the lake bottom was not sterile. It contained more cells than the water above. Bacteria living between sediment grains. Archaea metabolizing minerals in the mud. Organisms moving through spaces too small to see. The sediment is not a dead zone. It is the most biologically active part of the lake. Most of the ecosystem may exist in the mud, not in the water, hidden in spaces between particles, unreachable by sampling methods that only collect water. This inverts normal assumptions about lakes. On the surface, the water is where life thrives. Fish swim, plankton drift, plants grow in shallow areas. The sediment is seen as lifeless mud. But here the sediment is where life persists. The water is nearly empty by comparison. The real ecosystem is below the lake floor. In darkness, under pressure, unobserved, heat rises from the Earth's interior. This is true everywhere on the planet. Radioactive decay in the mantle generates energy. That energy moves slowly toward the surface. In most places, the heat flux is low, around 60 milliwatts per square meter, barely enough to notice. But in certain regions, the heat flux is higher. Volcanic zones, rift valleys, regions where the crust is thin. Parts of Antarctica have elevated geothermal heat, especially in West Antarctica, where the crust is stretched and thin. The heat reaches the base of the ice sheet. It melts ice from below. It warms the bedrock, and it enters the subglacial lakes. But the warming is minimal. The water temperature does not rise above freezing. It remains at -1 or -2 Celsius. The heat is not enough to warm the lake, only enough to prevent further freezing. For organisms, this heat represents energy, not thermal energy they can use for warmth, but chemical energy released by reactions between heated rock and water. When geothermal heat warms, bedrock's certain minerals become more reactive. Iron oxidizes. Sulfur compounds dissolve. Hydrogen gas is released. These reactions are slow, but they are continuous, and they provide a steady supply of chemical fuel for microbial metabolism. The organisms do not experience the heat as warmth. They experience it as a gradient in chemical potential, a difference between reduced compounds near the bedrock and oxidized compounds in the water above. That gradient is what they exploit. They position themselves at boundaries where both types of compounds are available. They move electrons from one compound to another. The energy released is captured, used to build cellular components, to repair damage, to replicate slowly when resources allow. The process is chemosynthesis, the same process that supports life at deep-sea vents. But slower, much slower. At near freezing temperatures, every chemical reaction takes longer. Enzymes work less efficiently. Diffusion rates decrease. The entire metabolism slows to a pace that seems almost geological. Doubling times measured not in hours but in weeks. Growth rates barely detectable, yet enough to sustain populations over millions of years. Once an organism enters a subglacial lake, it cannot leave. There is no migration pathway, no seasonal movement to other habitats, no dispersal of offspring to distant locations. The lake is a closed system, bounded by ice on all sides. The only exit is through outflow channels that lead to other subglacial lakes. But movement through these channels is passive. An organism cannot swim against the current. It drifts if the water carries it. If the water is still, the organism remains, generation after generation in the same location. This creates evolutionary constraints that do not exist in surface environments. On the surface, organisms can disperse. Seeds blow in the wind. Spores travel in water. Animals migrate. Genes flow between populations. This gene flow prevents excessive specialization. It maintains genetic diversity. It allows populations to adapt to changing conditions. But in subglacial lakes, there is no gene flow. Each lake is isolated from others. Each population evolves independently. Mutations accumulate. Some are beneficial in this specific environment. Those spread through the population. Others are neutral or harmful. Those are eliminated or persist at low frequency. Over millions of years, the genetic differences between lake populations become substantial. Organisms in Lake Vostok are likely genetically distinct from organisms in Lake Whillans, even if they descended from the same ancestral species. They have diverged, each adapted to the precise conditions of their particular lake. The temperature, the chemistry, the pressure, the nutrient availability. Every variable is different between lakes, and every population reflects those differences. This is evolution without escape, adaptation without the possibility of leaving. The lake is not a habitat. It is a trap, and everything living there is the descendant of organisms that could not leave. At the bottom of the ice sheet, something strange happens. Water freezes onto the underside of the ice. This is not normal freezing. On the surface, water freezes when heat is lost to the air. The top layer becomes solid first. Ice forms downward. But at the base of the ice sheet, the process reverses. Water in the lake is supercooled by surface standards. But it is liquid because of the pressure. When that water contacts ice above, the pressure drops slightly. The melting point changes. The water freezes. Ice grows upward, accreting onto the bottom of the ice sheet. This process is slow, millimeters per year. But over thousands of years, the accretion is significant. Some of the ice in the Antarctic ice sheet did not fall as snow. It formed from lake water freezing upward. This ice is different. It contains no air bubbles. Snow traps air as it compresses into ice. But water freezing from below does not trap air. It forms clear solid ice. In ice cores, this difference is visible. Above a certain depth, the ice contains tiny bubbles. Below that depth, the ice is clear. The transition marks the boundary. Above is ice that fell as snow. Below is ice that froze from the lake. This upward growing ice carries traces of the lake water, dissolved gases, chemical isotopes, sometimes fragments of microbial DNA. These traces are fragmentary. The DNA is degraded. The chemical signals are faint, but they are evidence, proof that the lake below contains life. or contained life at some point in the past. Whether that life still exists now is unknown. The ice preserves history, not current conditions. The water does not move in ways you would recognize as movement. There are no waves, no turbulence, no visible currents. The surface of the lake is pressed against solid ice, frozen in contact. Below the surface, the water exists in layers, each layer slightly different in density, slightly different in temperature. The differences are measured in thousandths of a degree, but they are enough to prevent mixing. Cold water near the ice, slightly less cold water deeper down. Where geothermal heat enters from below the water is fractionally warmer, still below freezing by surface measurements, but warmer relative to the layers above. This warmth does not rise quickly. Convection is weak. The density differences are minimal. Water moves upward in flows too slow to detect. Centimeters per year, perhaps less. A parcel of water beginning at the lake bottom might take thousands of years to reach the ice above. By the time it arrives, conditions at the bottom may have changed, or they may be identical. There is no way to know. The stillness is not emptiness. Particles drift in the water. Mineral grains too small to settle quickly. Fragments of rock ground to powder by the ice sheet. Organic molecules produced by microbial metabolism. These particles move with the water, rising or sinking depending on density, drifting laterally in currents too weak to measure. Their paths are determined by forces invisible at human scales. Brownian motion, molecular collisions, random walks through three-dimensional space. Over years, a particle might travel meters. Over millennia, it crosses the lake. Nothing rushes. Nothing needs to. The bacteria in the water column exist at densities difficult to imagine. In surface, lakes' microbial densities are high. Millions of cells per milliliter. Blooms of algae. Clouds of bacteria feeding on organic matter. The water is thick with life. But in subglacial lakes, the density is lower, by orders of magnitude. In Lake Willen's samples showed roughly 100,000 cells per milliliter. That sounds substantial, but it is sparse compared to surface water. The cells are spread through enormous volume. In a lake with volume measured in cubic kilometers, even billions of cells represent a diffuse population. Each cell occupies a space far larger than itself. The distance to the nearest neighbor might be millimeters. In microbial terms, that is vast. Chemical signals do not travel easily across such distances. Nutrients released by one cell may never reach another. Cooperation between cells becomes difficult. Each organism functions in near isolation. Not complete isolation, but functional isolation, separated by cold water that slows diffusion, by pressure that limits movement, by darkness that prevents signaling through light. The cells do not form mats. They do not cluster. They drift alone, metabolizing slowly, dividing rarely, waiting. The waiting implies expectation, and there is nothing to expect. No seasonal bloom, no influx of nutrients, no change in conditions that would favor growth. The environment is stable, and stability means each cell exists in the same state indefinitely. Active but not growing, alive but not expanding, persistent without progress. Energy is limited. There is no sunlight, no organic carbon washing in from terrestrial ecosystems, no dead plankton raining down from productive surface waters. The only energy comes from chemistry, reactions between minerals and water, oxidation of reduced compounds, reduction of oxidized compounds. The energy yield is small. A cell might harvest only a few molecules of ATP from a single reaction cycle. ATP is the energy currency of life. Each molecule represents a small amount of usable energy. In surface organisms, ATP is produced rapidly. Photosynthesis generates ATP continuously in the presence of light. Respiration generates ATP continuously when oxygen is available. A surface bacterium might produce millions of ATP molecules per second. But in subglacial lakes, production rates are far lower, perhaps thousands of ATP molecules per second, perhaps fewer. The cell uses this energy sparingly. Protein synthesis is slow. DNA replication happens infrequently. Movement is minimized. Some bacteria have flagella, but rarely use them. Swimming consumes energy that cannot easily be replaced. Better to drift, to conserve, to maintain only the essential processes. Membrane integrity, ion gradients, DNA repair. Everything else is optional. The cell exists in a state of enforced minimalism. Not by choice, by necessity. The environment provides just enough energy to prevent death, but not enough to support rapid growth. The population is maintained, not expanded. Generation times stretch. Weeks between divisions. Perhaps months. In some cases, years. A bacterial cell that divided once per year would seem absurd in a surface environment. But here it is normal, even optimal. Dividing faster would exhaust available resources, would require energy that does not exist. Slowness is not a disadvantage. It is the only viable strategy. Iron is common in bedrock. It exists in reduced form. Ferrous iron. 2 electrons in the outer shell. Chemically reactive. Easily oxidized. When water contacts iron-bearing rock, the iron dissolves. Slowly, it enters the water as ferrous ions. These ions are invisible. The water appears clear, but the iron is present, moving through the water column, rising toward the ice above. As it rises, pressure decreases. Temperature decreases. The chemistry changes. In the presence of oxygen, ferrous iron oxidizes to ferric iron. 3 electrons in the outer shell. Less reactive. Less soluble. Ferric iron precipitates out of solution. Forms tiny particles of rust. Iron oxide. These particles settle slowly back toward the lake bottom. The cycle completes. Iron rises as dissolved ions. Iron falls as solid particles. The time scale is geological. A single iron atom might complete this cycle once every 1,000 years, or once every 10,000 years. The rate is irrelevant. The cycle continues regardless. For microorganisms, this cycle is food. Not food in the sense of organic matter, but food in the sense of available energy. Some bacteria oxidize ferrous iron. They take the electron and use it to generate ATP. The iron becomes ferric, precipitates, falls. Other bacteria reduce ferric iron back to ferrous. They donate an electron. Use the reaction to generate ATP. The iron dissolves again. Rises. Between these two processes, the iron cycles. And energy is extracted at each transformation. Not much energy, but enough. The ecosystem is built on this cycling. Iron oxidizers near the surface where oxygen is slightly more available. Iron reducers in the sediment where oxygen is absent. Between them a flow of iron atoms. Up and down. Dissolved and solid. Reduced and oxidized. The lake is a battery. Chemical potential moving between states. And life occupies the circuit. Lake Vostok is 250 kilometers long. The water at the southern end is not the same as the water at the northern end. Temperature varies by fractions of a degree. Chemistry varies slightly. Depth varies significantly. At the southern end, the lake is shallow, a few hundred meters deep. The bedrock rises toward the surface. Geothermal heat flux is lower. At the northern end, the lake is deep, over 1,000 meters. The bedrock drops away into a basin. Geothermal heat flux is higher. Between these two ends, conditions grade continuously, not in discrete zones, but as a gradient. Organisms at one end of the lake are adapted to conditions there. Slightly different temperature, slightly different pressure, slightly different availability of nutrients. Organisms at the other end are adapted differently. The genetic differences might be subtle. Single mutations in key enzymes, variations in membrane lipids, small changes that allow function in specific conditions. But over the length of the lake, these small changes accumulate. The population is not uniform. It is structured by geography. Distance matters, not because organisms cannot disperse, but because dispersion is so slow that it becomes irrelevant. A bacterium at the southern end of the lake will never reach the northern end. Not in its lifetime. Not in 1,000 lifetimes. The water moves too slowly. The distance is too great. Each location in the lake is effectively isolated from distant locations. The lake is not one habitat. It is hundreds of microhabitats, each slightly different, each occupied by populations that do not mix. At the bottom of the lake, the sediment is layered, not in the way surface lake sediments are layered. There are no seasonal bands, no pollen zones marking climate changes, no ash layers from volcanic eruptions. The layering is chemical. Different minerals at different depths, different oxidation states, different microbial populations. The top layer is oxygenated. What little oxygen exists in the lake water penetrates a few centimeters into the sediment. Aerobic bacteria live here. They use oxygen, produce carbon dioxide, oxidize organic molecules if any are present. Below this, the oxygen is gone, consumed by the organisms above, never replaced. Here, anaerobic organisms dominate. They do not tolerate oxygen. They use other compounds as electron acceptors. Nitrate, sulfate, iron oxides. Each compound is reduced in sequence as you go deeper. Nitrate is reduced first. Organisms produce nitrogen gas. It bubbles up slowly through the sediment, dissolves in the water, rises toward the ice. Below the nitrate zone, sulfate is reduced. Organisms produce hydrogen sulfide. The sediment smells of decay, though nothing is decaying. The smell is chemical. Sulfur compounds released by metabolism. Below the sulfate zone, iron is reduced. The sediment turns black. Iron sulfide precipitates. Forms tiny crystals between grains. At the very bottom, methane is produced. Methanogenic archaea. They reduce carbon dioxide to methane. The simplest metabolism. The lowest energy yield. The last organisms in the sequence. Below them, nothing. Just rock. Cold. Sterile. Unweathered by biology. Each layer is a niche. Each occupied by specialists. No organism can move between layers. The chemistry is too different. An aerobic bacterium placed in the methanogenic zone would die. A methanogen brought to the surface would be poisoned by oxygen. The vertical structure is rigid, enforced by chemistry, stable over timescales longer than ice ages. Sound does not travel far in water this cold. The speed of sound decreases with temperature. At -2°C, sound moves roughly 1,300 meters per second through water. That is slower than at higher temperatures. But the speed itself is not the issue. The issue is attenuation. Sound waves lose energy as they travel. In warm water with suspended particles, an organism's attenuation is high. The water is noisy with biological sounds. Snapping shrimp, whale calls, fish vocalizations. But in subglacial lakes, there are no large organisms, no animals, nothing that produces sound intentionally. The only sounds are geological. Ice cracking far above. The pressure release sounds like distant thunder. Low frequency. Traveling through the ice more than through the water. Occasionally bedrock shifts. Micro-earthquakes. Grinding movements along fault lines. These produce vibrations, but infrequent. Years between events. The rest is silence. Not the silence of an empty room. The silence of a space where sound has no source. Bacteria do not make noise. Water that does not flow makes no sound. Sediment settling makes no sound. Chemical reactions are silent. If you could place a microphone in the lake, it would record almost nothing. Background thermal noise. Random molecular vibrations. But no signal. No pattern. No information. The lake exists in a state beyond silence, where silence is not the absence of sound, but the absence of any source that could produce sound. Radioactive isotopes in the water decay at known rates. Carbon-14, tritium, chlorine-36. Each isotope has a half-life, a predictable rate of decay. By measuring the ratio of parent isotope to daughter isotope, you can estimate age. How long the water has been isolated from the atmosphere. In surface water, these isotopes are replenished. Cosmic rays produce carbon-14 in the atmosphere. It dissolves in rainwater, enters lakes and oceans. The concentration stays relatively constant. But in subglacial lakes, there is no replenishment. The water was isolated when the ice sheet sealed the lake. Since then, the isotopes have been decaying. Carbon-14 has a half-life of 5,700 years. After 15 million years of isolation, the carbon-14 is gone. Completely decayed. Undetectable. Chlorine-36 has a half-life of 300,000 years. After 15 million years, very little remains. The water is isotopically dead. It carries no signature of recent atmospheric contact, no trace of modern carbon, nothing that would indicate exchange with the surface world. This makes age dating difficult. Without radioactive isotopes, you cannot measure time directly. You can only estimate based on ice dynamics. How fast the ice sheet moved, how long it took to reach its current thickness. These estimates are uncertain. The ice may have thickened faster in some periods, slower in others. Climate variations affect accumulation rates. The age of isolation is a range, not a number. Somewhere between 10 and 20 million years. Perhaps longer. The precision does not exist. Time becomes approximate, measured in millions of years plus or minus millions of years. At these scales, time stops functioning as a useful variable. Events cannot be ordered chronologically. Cause and effect become ambiguous. The water is old. That is certain. How old is unknowable with current methods. Where the lake water meets the ice above a boundary layer forms a transition zone, a few meters thick. In this zone, the temperature gradient is steep. The water is coldest at the ice contact, exactly at the freezing point for the local pressure. A few meters below the water is slightly warmer, hundredths of a degree warmer, but enough to prevent freezing. Within this boundary layer, strange things happen. Ice crystals form and dissolve continuously. The water is supersaturated with dissolved gases. As ice freezes onto the ceiling, gases are excluded. They concentrate in the boundary layer. The water becomes slightly more saline, more dense. It sinks away from the ice, replaced by water rising from below. This creates a very slow circulation, entirely confined to the boundary layer. The water below is unaffected. For organisms, this boundary layer is unique habitat, different from the open water, different from the sediment. The chemistry changes rapidly across a short distance. Gradients in temperature, gradients in dissolved gases, gradients in salinity. Some microorganisms may specialize in this zone, positioned where multiple resources are available, where energy can be extracted from the chemical differences. But the boundary layer is inaccessible. No sampling method can reach it without drilling through the ice. And drilling would destroy it. The pressure release would cause explosive degassing. The temperature change would disrupt the gradients. What exists there remains theoretical, inferred from models, from understanding of physical chemistry, but not observed, not confirmed. The boundary layer is a hypothesis, well supported, but untested. Photosynthesis does not occur in these lakes. This is obvious but worth stating clearly. Every food chain you are familiar with begins with photosynthesis. Plants capture sunlight, convert it to chemical energy, store it in sugars and starches. Herbivores eat the plants. Carnivores eat the herbivores. Energy flows from light through organisms in sequence. But here there is no light. No photosynthesis. No primary production in the traditional sense. The base of the food chain is not plants. It is chemosynthesis. Bacteria that oxidize inorganic compounds. Iron, sulfur, ammonia, hydrogen. These organisms are primary producers. But they do not produce much. The energy available from chemical reactions is far less than the energy available from sunlight. A photosynthetic cell can produce thousands of times more energy than a chemosynthetic cell. This limits everything above it. If primary production is low, secondary production must be lower. There are no herbivores in subglacial lakes. Nothing that grazes on bacteria. Nothing large enough to consume cells. The only consumers are other bacteria, species that break down the waste products of chemosynthesizers, that scavenge dead cells, that recycle the limited organic carbon. The food web is microbial, entirely microbial. Bacteria and archaea. Nothing else. No protozoans. No fungi. No multicellular organisms of any kind. Life exists at the simplest organizational level. Single cells, each functioning independently. No cooperation. No predation. No symbiosis. Just metabolism. Slow. Efficient. Minimal. The deepest parts of Lake Vostok have never been sampled. They may never be sampled. The technical challenges are prohibitive. The ethical concerns are substantial. What exists in the deep basin is unknown. Water temperature there may be slightly warmer. Geothermal heat accumulates, rises through the bedrock, enters the water from below. If the temperature is warmer, chemical reaction rates increase. Microbial activity might be higher, or it might not. Warmer water is less dense. It should rise. But if the water is also more saline, density increases. Salt water is heavier. It stays at the bottom, a stable layer, unmixed, untouched by the water above. If this layer exists, it could be millions of years old, older than the lake itself. Water trapped when the ice sheet first formed, never exchanged, never replaced, sealed at the bottom of a basin under kilometers of ice. The chemistry in this layer would be unusual. Oxygen completely absent. Dissolved minerals at high concentration. Hydrogen sulfide. Methane. Ammonia. Conditions that are toxic to most life. But some organisms thrive in these conditions. Extremophiles. Species adapted to environments that seem incompatible with biology. They might exist in the deep basin, metabolizing slowly, reproducing rarely, constituting an ecosystem completely separated from the water above. But this is speculation, educated speculation based on physical principles, but speculation nonetheless. The deep basin has not been observed. It cannot be observed without destroying what makes it unique. It remains a blank space, a region of complete uncertainty. Some cells in the lake may not be active. They may be dormant, in a state of suspended animation. Metabolism reduced to nearly zero. No growth, no division, no response to environment, just survival. In surface environments, dormancy is common. Seeds remain viable for years. Bacterial spores survive centuries. Tardigrades endure decades in stasis. Dormancy is a strategy for surviving unfavorable conditions, waiting for conditions to improve. But in subglacial lakes, conditions do not improve. They do not worsen either. They simply remain constant. So why would an organism enter dormancy? Perhaps it has no choice. Perhaps nutrient levels occasionally drop below the threshold needed for active metabolism. The cell shuts down, enters a minimal state, waits for nutrients to increase. Days pass, weeks, years. Eventually, a pulse of nutrients arrives, carried by slow currents, or released by other cells. The dormant cell activates, resumes metabolism, grows briefly, divides if possible, then returns to dormancy. This cycle could repeat thousands of times over millions of years. Each period of activity brief, each period of dormancy long. The cell is alive in the technical sense, but alive in a way that barely resembles active life. More like a suspended state. Existence without experience. Persistence without awareness. If cells can be aware, which they cannot, but the metaphor holds. Dormancy is existence reduced to its minimum. And in subglacial lakes, minimum may be the norm. Lake Vostok is connected to other lakes downstream. Water exits through channels carved in bedrock. The flow is slow, perhaps a few meters per year. Over time, water moves from one lake to the next, carrying dissolved minerals, carrying microorganisms, carrying genetic material. But the transit time is enormous. A water parcel leaving Lake Vostok might take 10,000 years to reach the next lake. Any organism in that water would experience the journey as environment, not as movement. The channel is habitat. Dark, narrow, high pressure, water flowing too slowly to generate turbulence. Organisms in the channel would be adapted to channel conditions, different from lake conditions, different from sediment conditions. The channel connects lakes, but it also separates them. The distance is too great for rapid genetic exchange. By the time an organism travels from one lake to another, mutations have accumulated. The organism may no longer be the same species that entered the channel. Speciation occurs in transit. The channel is not a corridor. It is an environment in itself, with its own selection pressures, its own isolated populations. The network of subglacial lakes is not one system. It is many systems, loosely connected, exchanging material on timescale longer than evolutionary change. Each lake is unique. Each channel is unique. The whole forms a mosaic, patches of habitat separated by distance that functionally equals isolation. Videos like this take time to create. If they help you relax, subscribing helps the channel continue. Pressure affects every molecule in a cell. At 300 atmospheres, proteins compress. Lipids pack more tightly. DNA helixes shorten slightly. Water itself becomes more dense. Its molecular structure changes. Hydrogen bonds compress. The cells in subglacial lakes have adapted to this. Their proteins are different from surface proteins. More flexible in key regions, more rigid in others. The amino acid sequences are modified. Mutations that would be harmful at surface pressure are beneficial here. The proteins fold into shapes that only work under compression. If you brought these cells to the surface, they would not survive. The pressure release would cause proteins to unfold. Membranes would become too fluid. Cellular machinery would fail. The organism would die. Not from heat, not from oxygen, from the simple absence of pressure. They are obligate barophiles, pressure-loving, pressure-requiring. They cannot exist without it. This is adaptation at the molecular level. Every enzyme, every membrane protein, every ribosome, shaped by millions of years under compression. The cells are not visiting this pressure. They are made of it, built by it, dependent on it. Pressure is not external force. It is internal structure. Some of the metabolic pathways used by organisms in subglacial lakes are not fully understood. They produce compounds that do not fit known biochemical cycles. Unusual lipids, unidentified organic acids, molecules that suggest reactions not documented in surface organisms. This may be analytical error, contamination, misidentification, or it may be real. Novel biochemistry, metabolic pathways that evolved in isolation, that have no analog in surface life. The compounds are rare, present in trace amounts, difficult to analyze, difficult to replicate. Labs have tried to culture organisms from subglacial lake samples. Most attempts fail. The organisms do not grow at surface pressure, or surface temperature, or with available nutrients. They remain dormant, or they die. A few attempts have succeeded. Slow-growing colonies, taking weeks to become visible. But even these do not behave normally. They produce unexpected waste products. They consume nutrients at unusual ratios. They respond to stimuli in ways that suggest different metabolic logic. Whether this represents true novelty or simply extreme adaptation is unclear. The data is insufficient. The samples are too degraded. The analysis is too limited. What exists in the lakes may be familiar life pushed to extremes. Or it may be something genuinely different. Life that took a divergent path, that explored chemical space unavailable to surface organisms, that found solutions to problems that do not exist elsewhere. The question remains open. Nothing changes quickly here. The temperature remains. The pressure remains. The darkness remains. 1,000 years is not long enough to measure change. 10,000 years might show minor variations. Chemical concentrations shifting slightly. Sediment accumulating by millimeters. Ice accreting grain by grain onto the ceiling. But these are not events. They are conditions continuing. The lake does not develop. It does not progress toward any state. It simply is, and has been, and will be, for as long as the ice above remains. The organisms within reflect this. Their generation times are long. But even summed across millions of generations, the change is minimal. Adaptation to conditions that do not vary. Refinement of efficiency in an environment that provides no surplus. The genome of a bacterium in Lake Vostok today may differ from its ancestor 1 million years ago. But the differences are subtle. A few genes added, a few lost. Regulatory regions modified. Nothing dramatic. No new body plans. No complexity gained. Just persistence. Optimized slowly across time that has no meaning to the organisms experiencing it. Lake Vostok is not unique in the solar system. There are other worlds with subsurface oceans. Europa, a moon of Jupiter, covered entirely in ice. The ice layer is kilometers thick. Beneath it is liquid water, an ocean larger than all of Earth's oceans combined. Heated by tidal flexing, Jupiter's gravity pulls on Europa. The moon deforms. The interior heats. Ice at the base melts. The conditions are similar to Antarctic subglacial lakes. Darkness, high pressure, chemical energy from rock-water interactions. If life exists in Lake Vostok, it suggests life could exist on Europa. The same principles would apply. Chemosynthesis, slow metabolism, microbial ecosystems sustained by geology rather than sunlight. Enceladus is another candidate. A moon of Saturn, ice-covered. Geysers erupt from cracks in the southern hemisphere. Water vapor and organic molecules shoot into space. The plumes suggest a subsurface ocean, warm enough to remain liquid, active enough to drive circulation. The chemistry detected in the plumes includes ammonia, methane, hydrogen, the building blocks of chemosynthetic metabolism. Whether life exists there is unknown, but the conditions are not impossible. They are familiar, at least to organisms that inhabit subglacial lakes. The discovery of life beneath Antarctic ice makes life elsewhere seem less unlikely. Not certain, but within the range of possibility. The universe may contain many sealed oceans, many dark lakes, many isolated ecosystems sustained without stars. No human has seen these lakes directly. No camera has recorded their waters. No instrument has measured their depths in real time. Everything known comes from inference. Radar reflections interpreted as water. Ice cores analyzed for chemical traces. Models constructed from physics and chemistry. The lakes exist in data, in graphs, in maps showing estimated boundaries, but not in images, not in footage, not in direct observation. This does not make them less real, only less familiar. Most of the universe exists this way, known through measurement rather than sight. Gravity detected by its effects on orbits. Dark matter inferred from galactic rotation. Subglacial lakes are no different. Their reality is mathematical, described by equations, confirmed by consistency across multiple measurement techniques. The water is there. The life within it is there, even if no eye has witnessed either, even if no eye ever does. The lakes do not require observation to exist. They preceded observation by millions of years. They will persist long after observation becomes impossible. When the ice sheet eventually melts, thousands of years from now, or hundreds of thousands, the lakes will drain. Water will flow to the ocean. The organisms within will be carried to new environments. Most will die, unable to adapt quickly enough, unable to tolerate the change in pressure, in temperature, in chemistry. But some may survive. Some may find niches in deep ocean sediments, in subsurface aquifers, in places that resemble the conditions they left. They will carry forward, descendants of lineages that spent millions of years in darkness, shaped by isolation, adapted to constancy, suddenly thrust into a world that moves and changes and offers no stability. Whether they persist there is unpredictable. How many species exist in Lake Vostok? Unknown. How do they reproduce? Unknown. How do they sense their environment? Unknown. Do they have predators? Unknown. Do they compete for resources? Presumably. But the mechanism is unknown. How long can a cell remain dormant and still revive? Unknown. What is the maximum depth at which life persists in the sediment? Unknown. Are there viruses in the lake? Probably, but unconfirmed. Do the viruses infect the bacteria? Presumably, but never observed. How does genetic material transfer between cells? Unknown. Is there horizontal gene transfer? Possibly, but the rate and mechanism are unknown. What proportion of the microbial community is culturable? Very small. Most organisms cannot be grown in laboratory conditions. They remain unidentified. Their metabolisms uncharacterized. Their roles in the ecosystem unresolved. The list of unknowns is longer than the list of knowns. This is normal in science. But here the unknowns feel more fundamental. Not gaps in data, but gaps in accessibility. The lakes cannot be studied without being altered. Every measurement changes what is being measured. Every sample removes something irreplaceable. The pristine nature of the environment is its defining feature, and also the barrier to understanding it. There are no fossils in subglacial lakes. Fossils require mineralization, replacement of organic material with stone. This happens in sediments where minerals are abundant and conditions are right. But in subglacial lakes, mineralization does not occur. The sediment chemistry is wrong. The time scales are wrong. When a cell dies, it decomposes, broken down by other microorganisms, recycled into nutrients. Nothing remains. No shell, no skeleton, no trace. The history of the lake is not preserved in its sediments. Each layer is chemically distinct, but biologically uniform. The same types of organisms at every depth. No succession, no progression, no record of evolutionary change. The past is invisible. Only the present exists. And the present is indistinguishable from 1,000 years ago, or 10,000, or 100,000. Without fossils, without isotopes, without any marker of change, time collapses into a single moment, extended indefinitely. Lake Vostok will remain sealed, not by decision, by necessity. The risk of contamination is too great. The value of the pristine state is too high. Drilling has stopped above the water. Instruments placed nearby monitor conditions remotely. But they do not penetrate. They do not sample. They do not disturb. Other lakes have been sampled. Lake Willans, Lake Mercer, smaller lakes where the scientific value of direct sampling outweighed the risk. But Lake Vostok remains untouched. A reference, a control, a place that represents what these environments are without human interference. Perhaps future technology will allow non-invasive study, sensors that can be placed without drilling, imaging techniques that can penetrate ice without disturbing water, DNA sequencing from minute samples of accreted ice. Or perhaps not. Perhaps some places remain permanently beyond reach. Not because the technology will never exist, but because accessing them destroys what makes them worth accessing. The lake is valuable because it is isolated. To study it is to end that isolation. The choice is between knowledge and preservation. Science has chosen preservation, at least for now. The bacteria in the lake are not waiting. Waiting implies anticipation, implies a future state that differs from the present. But there is no future state, only continuation. The cells metabolize. They maintain. They persist. Not because persistence leads anywhere, but because persistence is the only option. There is no goal, no purpose in the teleological sense, just chemistry, reactions proceeding because conditions allow them to proceed. Energy gradients exist. Organisms exploit those gradients. The exploitation releases energy. The energy sustains the organism. The cycle continues, not toward anything, not away from anything, simply continues. This is existence without narrative, without arc, without progression. The beginning was millions of years ago. The end will be millions of years from now. The middle is indistinguishable from either. The lake is in equilibrium, not static equilibrium, dynamic equilibrium. Process is balance. Ice melts at the base of the ice sheet. Ice accretes at the lake ceiling. The water volume remains constant. Water enters from upstream lakes. Water exits to downstream lakes. The chemistry remains stable. Minerals dissolve from bedrock. Minerals precipitate from water. The concentration stays within narrow bounds. Organisms are born. Organisms die. The population size does not change. Energy enters from geothermal heat. Energy is dissipated through metabolism and chemical reactions. The balance is maintained, not by regulation, not by feedback mechanisms, by the simple fact that any imbalance is immediately corrected. Too much growth depletes nutrients. Growth stops. Too much nutrient accumulation without growth is impossible. Organisms respond by growing. The system self-regulates through chemistry, not through biology. The organisms are participants, not controllers. The sun exists, above the ice, above the atmosphere, shining, but its light does not reach the lake. Its warmth does not reach the lake. Its cycles do not affect the lake. The lake is disconnected from the star that powers most of Earth's biosphere. This disconnection is absolute, not partial, not mediated, complete. The energy that sustains life in the lake comes from the planet itself, from radioactive decay deep in the mantle, from heat slowly conducted through rock, from chemical reactions between stone and water. Earth provides, not the sun. This inverts the normal relationship between star and life. On the surface, everything traces back to sunlight. Plants capture it. Animals eat the plants. Decomposers break down the animals. The cycle is solar-powered. But here the cycle is geological, powered by processes that would continue even if the sun vanished, even if the planet drifted into interstellar space. The lake would persist, for a time, until the planet's interior cooled, until geothermal heat stopped flowing. But that time scale is billions of years, longer than the remaining lifespan of the sun. The lake is more permanent than the star above it. The water in the lake has been water for a very long time. Liquid water, Not ice, not vapor. The molecules have not changed phase in millions of years. They have remained liquid under pressure, in darkness, at temperatures that would freeze them at the surface. But here they flow, slowly, imperceptibly, but flow nonetheless. The water is patient, not in the sense of waiting, but in the sense of continuing without requirement for change. Each molecule moves according to forces acting on it. Temperature gradients, pressure gradients, gravity. The movement is deterministic, not random, but so slow that randomness appears. Brownian motion, thermal fluctuations, paths that seem chaotic over short scales, but are ordered over long scales. The water will eventually exit the lake, flow downstream, reach the ocean, evaporate, return to the atmosphere, fall as snow, compress into ice, complete the cycle. But the time scale for this is geological. A single molecule might take 1,000,000 years to complete the journey. From atmosphere to ice to lake to ocean to atmosphere again. During that time, empires will rise and fall. Species will evolve and go extinct. Continents will shift position. But the water molecule remains water, following physical law. Unaware, unconcerned. Lake Vostok exists. This is certain. Life exists within it. This is probable. The life is microbial. This is almost certain. The ecosystem is chemosynthetic. This is consistent with available evidence. Beyond these statements, certainty fades. The number of species, unknown. The complexity of interactions, unknown. The history of the populations, unknown. The future of the ecosystem, unknown. What remains is the fact of existence. Water under ice, life in water, metabolism in darkness, persistence across time that exceeds human comprehension. The lake does not need to be understood to continue. It continued for millions of years before it was discovered. It will continue long after interest in it fades. The organisms within have no awareness of being studied, no response to being wondered about. They simply are, existing in conditions that seem extreme from the outside, but are normal from within. This is the final truth of subglacial lakes. They are environments, not mysteries, not enigmas, just places where different rules apply, where life found different solutions, where existence continues in forms that require no observation to validate them. This is only one of many hidden environments shaped by isolation. Across Antarctica, other lakes exist beneath the ice. Each separated, each unique, each containing ecosystems that have diverged in the darkness. Some lakes are larger than Vostok. Some are deeper. Some have been isolated longer. In the next exploration, the descent continues toward lakes that have remained sealed even longer than Vostok. Waters that last exchanged with the atmosphere before ice existed on Antarctica, before the continent was fully separated from other landmasses. Ecosystems that may preserve forms of life otherwise extinct. That is where the journey leads. The ice above remains, white, silent, kilometers thick. Beneath it the water remains, dark, still, cold but liquid. The boundary between them is precise. Ice on one side, water on the other. The boundary does not move, does not shift. It simply exists, separating two states of the same substance. One solid, one liquid, both containing traces of life, adapted to conditions at the edge of survival, or what seems like the edge from outside. From within, it is just existence. Normal, ongoing, unremarkable to the organisms experiencing it. The lake continues. The ice continues. The darkness continues. Nothing resolves. Nothing concludes. The water remains beneath the ice. The life remains within the water. The pressure remains constant. The temperature remains constant. Time passes without marking its passage. Years, centuries, millennia. The lake persists, unchanged, unobserved.